Enveloped viruses such as SARS-CoV-2 access host cells by membrane fusion through either the plasma membrane or an endosomal membrane. We haven't talked about that pathway yet, but that's what happens when that gets taken inside the cell. Activation of SARS-CoV-2 spike protein through proteolytic cleavage is required for host cell entry, so thank God it has the polybasic furin cleavage site, because otherwise it wouldn't enter very well at all. Therefore, an understanding of the envelope protein activation by proteolytic cleavage is central to gaining insights to viral pathogenesis and development of potential therapeutics. The receptor binding domain of SARS-CoV-2 spike protein presents the most critical feature of this protein, which is located at the C-terminus of the S1, that's the spike protein subunit, and binds to ACE2 with high affinity. Not just high, but ridiculously high. Not just ridiculously high, but higher to humans than to any other species. A little alien, just saying. What happens is this is the ACE2 receptor here, and the spike protein has to come in, and it has to find a way to bind into this, and this overall conformation, every little fold and wiggle, really important. Well, maybe with zinc in there, it's folded one way. Maybe with zinc out of there, it's folded a slightly different way. Maybe that has something to do with this, but it's just interesting to me that the zinc binding motif for this ACE2 receptor and the spike binding motif for the S1 subunit of SARS-2, it's the same spot. They're kind of directed at the same spot. So take your zinc. All right.